The next step is to figure out for any of these traits that we can have that get passed on from parents to children, how likely is it that a child is going to have one trait or another? So we know that mom and dad each pass on one version of that gene, and as you get those two genes from mom and dad, they combine to produce your genes, your genetics. And so you'll have a trait based on what you get from mom and what you get from dad and how those two are related to each other. So let's just think about possibilities here. Let's say that we have mom with, say, a genotype like this, a big A and a little a for some trait. And dad has, how about big A, big A. So this is dominant recessive. So that one is heterozygous. Mom is heterozygous. Just put a note here. Heterozygous. And dad's is big A, big A. So those two are the same. That makes this one homozygous. Homo for same. Homozygous. And they're both the big A's. Big A's go with dominant. Homozygous, dominant. Okay, so then the child. When mom and dad have a child, what does the child get? Well, there's a few different combinations that we have to look at here. So one possibility is that mom gives her big A and dad gives his big A here, and we end up with a child at big A, big A. So you get one of mom's and one of dad's. Mom and dad each have two, but you only get one from mom, and you get one from dad. So maybe they each give a big A. That means the child gets big A, big A. So homozygous, dominant. Another possibility, mom still gives her big A, and dad gives, well, another big, dad only has big A's to give, so he could give that other big A, and we'd still end up with the same genotype for the child. Big A, big A. Okay, third way this could work out, mom gives her little a, and dad gives this first big A. And so then we're going to have a little a and a big A. And the pattern that we use on this, we always just write the big A's first. The capital letters always come first. So it'll be big, little. That one is heterozygous, just like mom. And then last possibility on this one, mom gives her a little a, and dad gives this big A. And we end up again at one big and one little. So if we look at the child's possibilities, we have these two are the same. That is homozygous. So they're both the same, homozygous for same, and then dominant because it's all big A's. So that's just like dad. Or these two are the same. These are both heterozygous. Hetero meaning different. And notice that there's only 4 possible outcomes. 2 of them are over here, 2 of them are over here. So we'd say this will happen 50% of the time, and this will happen 50% of the time. So the child has a 50-50 chance, just like flipping a coin, of having genes of being homozygous dominant or heterozygous. Now, if this is a trait where all it takes is one big A to show the trait, the phenotype that's associated with having a big A, then this kid always gets at least one big A. This kid will always show the dominant phenotype. Now, This process of crossing over those different A's and figuring out the ratios, that's all we're doing with Punnett Squares too. We just organize it a little differently so it makes it a little bit faster and easier to keep track of everything. So let's set the same one up as Punnett Squares. So with a Punnett Square, we'll draw out a square that's made up of 4 smaller squares. So we draw a big square and divide it in half both ways. We'll put mom's information over on the side and dad's up here on the top. And so looking back, mom was heterozygous, so she's going to have one big A and one little a. And dad was homozygous dominant, so he had 2 big A's. Okay. And then the child, possibilities are all going to be inside the square here. So the child is going to be in red. And all we're going to do is, for each square, we're going to look at what kind of letter, capital or lowercase, shows up above that cell and what shows up to the left. The one above from dad, the one to the left from mom. So if I look in this cell here in the middle, I see that or on the left side up at the top rather, at the top we've got a big A from dad, and on the left side we've got a big A from mom. This one right here, at the top we've got a big A from dad, at the left side we've got a big A from mom. Down here, at the top we've got a big A from dad, at the left we've got a little a from mom. And this last one, at the top we have a big A from dad, at the left we have a little a from mom. Now if you notice back up here, that's exactly the same combinations that we got before. So these top 2 are big A, so just like these top 2 were big A, and then these bottom 2 are big A, little a, and these bottom 2 here are big A, little a. So it shows exactly the same combinations, it's just a little faster way to do it. And so we can say that here our child has 2 possible outcomes. So the genotypes for the child we've got big A, and that happens 2 out of 4 times, or 50% of the time. The other possibility is big A, little a, and that one also happens 2 out of 4, or 50% of the time. Last thing we can do here is think phenotypes for the child. So remember, the phenotype can be dominant or recessive, just like the genotype, the A's, genotypes. So the phenotype here is, do they get the dominant trait or the recessive trait? So if we're thinking about earwax, wet earwax is the dominant trait. Dry earwax is the recessive trait. But just in general here, both of those genotypes have at least one capital letter, at least one dominant allele, one dominant version of that gene. So here the only possibility is that we're going to have the child with the dominant phenotype. That'll happen 100% of the time. All 4 of those go with the dominant phenotype. Let's set up another one for practice. Let's say that mom has little a, and dad has big A, little a. Yeah, let's actually change that. Let's do big and little for each one. Big and little. There we go. So they both have a mixture now. They're both heterozygous for this trait. So if this is earwax, they both have wet earwax, but they also have the gene for dry earwax. Just the wet earwax is dominant. So we see the wet earwax as the result. So our Punnett square, get our square and divide it into 4. We're going to have dad up here, big A, little a, and mom over here, big A, little a. And then we'll fill in the information for the child. So in this square, we have a big A from dad, a big A from mom. We have big A, big A. Here, little a from dad, big A from mom. We always do the big A's first. Big A, little a. Here, big A from dad, little a from mom. Do the big A first, big A, little a. And here, little a from dad, little a from mom, little a, little a. So our genotypes, this time we've got 3 possibilities. We have big A, big A. That showed up out of these 4 boxes. It showed up only here. That's 1 out of 4 times, or 25%. We have big A, little a, that is heterozygous. And that showed up here and here. That's 2 out of 4 times, or 50%. And then last one that shows up in that table is little a, little a. That's right here. That also happened 1 out of 4 times, or 25%. As far as phenotypes, these two together have at least 1 copy of the dominant gene, 1 dominant allele. So those ones go with the dominant phenotype, so wet earwax if we're thinking in that example. The dominant. And because of these numbers here, we have 1 out of 4 times and 2 out of 4 times. Altogether, that's 3 out of 4 times. Or, as a percentage, 25% and 50% gives us 75%. Or this one, we don't have any copies of the dominant gene, so this one is going to be the recessive phenotype, dry earwax in that example. That only happens 1 out of 4 times, or 25%. Okay, at this point now you're ready for some practice.